Hello, this video introduces our CVPR 2025 highlight work titled DAMM-Diffusion, Learning Divergence-Aware Multi-Modal Diffusion Model for Nanoparticle Distribution Prediction. Nanoparticles, specifically nanoprobes, have become powerful tools in oncology, offering both diagnostic and therapeutic functions. These nanoscale structures can be precisely engineered to target tumors, enabling accurate imaging and localized drug delivery. This concept is closely tied to the tumor microenvironment, which is not only composed of tumor cells, but also includes immune cells, blood vessels, and the extracellular matrix. Importantly, tumors often feature abnormal and leaky vasculature, as well as impaired lymphatic drainage. These structural abnormalities are critical to a mechanism known as the EPR effect, or enhanced permeability and retention. As illustrated on the right, the EPR effect allows nanoprobes to passively accumulate in tumor tissue far more than in healthy tissues. Because of the leaky blood vessels and limited lymphatic clearance, therapeutic agents delivered via nanoparticles can remain in tumors longer, improving treatment efficacy. While the EPR effect provides a promising theoretical basis for nanoparticle-based drug delivery, its clinical translation remains a major challenge. One possible reason is the substantial heterogeneity of the EPR effect across individuals. It means that the distribution of nanoparticles can vary dramatically from one subject to another. These findings highlight the urgent need to better model and predict intratumoral nanoparticle distribution. To understand the intratumoral distribution of nanoparticles, we first consider two critical components of the tumor microenvironment: tumor vessels and cell density. These two factors greatly influence how nanoparticles accumulate and spread. However, the relationship between these structural features and the final nanoparticle distribution is highly complex and nonlinear, making it extremely challenging to model through traditional analytical approaches. So how to model this complex relationship? To tackle this problem, we start by leveraging image-based data that reflect the tumor microenvironment. Specifically, we use CD31 immunofluorescence to capture the structure of tumor blood vessels and DARPI staining to visualize nuclei. To obtain the ground truth for nanoparticle accumulation, we use QD's fluorescence imaging, where quantum dots, fluorescent nanoparticles, are tracked in tissue sections. These images serve as our multimodal input features. We then input this image data into a neural network. By learning from paired image and distribution data, the model is trained to predict how nanoparticles distribute among the tumor microenvironment. Here, we can formulate our task. Given multimodal tumor microenvironment components, such as vessel and nuclei images, our goal is to use neural networks to predict the intratumoral distribution of nanoparticles. Previous studies have explored how to model nanoparticle distribution based on tumor microenvironment images. These works highlight the potential of using deep learning on image-based multimodal data to capture complex biological patterns. However, key challenges still remain, particularly in handling divergence among multimodal data. While multimodal fusion can potentially enhance prediction performance, it also comes with challenges. In some cases, conflicting or noisy information from auxiliary modalities may actually degrade the model's output. For example, when the auxiliary modality introduces noise or misaligned features, the unimodal model might actually perform better than the multimodal method. In our task, tumor vessels are the dominant signal, as it is directly related to nanoparticle retention and transport. Nuclei images, on the other hand, serve as an auxiliary modality. They may offer additional spatial cues but also introduce inconsistent results. To balance these effects, our DAMM-Diffusion considers both unimodal and multimodal branches for NP's distribution prediction by considering the divergence among nuclei and vessels channels. Here is an overview of our proposed DAMM-Diffusion framework. You can see that it consists of two parallel branches, a unimodal branch that relies solely on vessel information, and a multimodal branch that incorporates both vessel and nuclear data. At each step of the reverse process, both the unimodal branch and multimodal branch perform the reverse step in a unified network and output the predictions respectively. The DAMM module is proposed to decide whether to apply multimodal predictions or only using the unimodal generation results for NP's distribution prediction. Next, let's explain the details of our divergence-aware multimodal diffusion models for NP's distribution prediction. Following the standard design of diffusion models, our framework consists of a forward process and a reverse process. In the forward process, the T-step forward process progressively adds noise to the original NP's distribution. In the reverse process, the unimodal and multimodal branches perform their reverse steps, conditioned on unimodal and multimodal data, respectively. The objective for each branch is derived by maximizing the evidence lower bound of the diffusion process. The Multimodal Fusion Module, or MMFM, is designed to effectively combine features from vessel and nuclear modalities. MMFM firstly extracts the useful spatial information from individual modality and then re-weights the channels after concatenating them together. Finally, the one-by-one convolution is applied to compress the channels. Besides the MMFM modules applied in the multibranch of our model, we develop an uncertainty-aware module, UAFM, that can calculate the uncertainty map for multimodal fusion during cross-attention computation. In our model, we treat the process of cross-attention as a bipartite graph, which can be divided into two disjoint and independent sets, nuclei set and vessel set. Each pixel feature is treated as a node, and attention weights define the edges across these sets. However, since most biological studies indicate that the NP distribution correlates strongly with vessels, we are uncertain if the auxiliary modality can help further improve the prediction performance, for example, pixel noisy or lacking essential information. Accordingly, the connections between the nuclei and vessel nodes may be unreliable for the combination prediction of NP via cross-attention. To address this, we introduce the concept of node uncertainty on the auxiliary modality nuclei set to measure the connection reliability to primary modality. The uncertainty-aware cross-attention assigns the uncertainty value to each node of the auxiliary modality and recompute the uncertainty-aware cross-attention weights by multiplying the edges weights and the uncertainty of the node. The existing study indicates that the divergence among multimodal data may lead to unreliable results when integrating them together. We propose a divergence-aware multimodal predictor, DAMM, to assess the consistency within the multimodal data. Specifically, we calculate the mean value of U, denoted as D, to represent the divergence for the current multimodal data. We just optimize the unimodal loss if D is larger to the threshold gamma. By contrast, we encourage multimodal fusion when the divergence is lower than a threshold gamma. To enhance the model's adaptability on gamma, we dynamically feed the results of multimodal fusion back to the divergence D and design a divergence feedback loss. We design a criterion as E to represent the effectiveness of multimodal fusion. Then the DFL is defined as the binary cross-entropy loss with a regularization term. In the prediction stage, the final output is selected conditioned on the divergence D, whether higher than the gamma or not. To evaluate the performance of our DAMM-Diffusion, we conduct cross-validation for internal validation and further test our model on the external validation. For the internal validation, a total of 25,097 patches are collected, and a 6-fold cross-validation is performed, with each fold corresponding to an individual T1 mice model. The external validation dataset is consisted of 3,800 patches derived from a different B16 tumor model in order to further validate the generalization ability of our model. We compare our model with several state-of-the-art unimodal and multimodal generative methods. As shown in the tables, our model consistently outperforms all baselines in terms of SSIM and PSNR on both internal and external sets. Additionally, we can observe that SOTA unimodal methods such as LDM and BBDM are superior to several multimodal generative methods like Diamond-GAN and GANDA. One possible reason is that these multimodal algorithms overlook the divergence among different modalities, leading to the inconsistent fusion results. We also conduct ablation studies to assess the impact of different components in our framework. We evaluate different image types in the unimodal branch. The results clearly show that using vessel images yields higher SSIM and PSNR than nuclei images, confirming that vessels are more predictive for nanoparticle distribution. Additionally, we present ablation results for MMFM, UAFM, and DAMMP. We find that each component contributes to performance improvement, and the full model achieves the best results, demonstrating the necessity of all three modules. We present the visualization of the generated NP's distribution and corresponding difference maps at patch level. As shown in the figure, our DAMM-Diffusion is effective in preserving smooth boundaries and maintaining the NP's distribution at patch level. Finally, the whole slide level predictions show that the NP's distribution predicted by our DAMM-Diffusion is closer to the ground truth than the comparing method. That's all of our video. Thanks for your listening.